First of all, thank you very much for giving us the opportunity to present Maieutix here. I introduce myself. My name is Oliver Carrillo. I'm co-founder of Maieutix, and I'm one of his software developers. And well, what Maieutix is, let's see if I can match here. What Maieutix is, well, we are a complementary and interdisciplinary team of biologists and computational scientists that offer computational biology and molecular dynamic simulation services to pharma companies that they not have a computational team by their own. Well, we want to act as a contract research organization that just from the in the preclinical stage, from the very beginning of the phase discovery, and we combine our proprietary algorithm, commercially available software, and open web resources and free databases. Well, what are the services we offer? Well, we offer, as we said before, simulation services. Actually, Monte Carlo dynamics and molecular dynamics services, which, as you may know, they keep the temperature, salinity, and the pH and the flexibility of the macromolecule kept. What are our applications priority? Well, first of all, drug discovery by applying binding mode analysis, in silico compound screening, toxicology, and binding kinetics analysis. And on the second hand, drug development by using of perturbation analysis, studies of the stability of temperature and pH, extractables and leachables interaction studies. Well, our business proposition, as you already know, drug discovery is expensive, it's risky, it's lengthy time-wise. What we propose is in silico services. That, well, in which we offer a state-of-art algorithm that we have access to innovation. So it's important for the new things that are coming to be to have a hand in the innovation purposes. And well, the advantages is that we increase the rate of the preclinical candidate selection, the increase of the therapeuticals. In this solution, we will diminish the time of compound screening and the time to reach the patient. Okay, let me say some words about our first software we developed in the group. It is called EDMC Algorithm. EDMC stands for Essential Dynamic Monte Carlo. As we said before, we are interested in drug discovery. In the discovery, it's need to, well, we need to respect and preserve the global flexibility of the macromolecule, and essential dynamic is important to achieve this objective. And we simulate complex biological systems and small molecules, such as, for example, a protein and a ligand, two proteins and a small molecule, or a DNA and small molecule. The flow chart we follow is, well, we start with a protein X-ray structure and ligand Smiles code, and we combine it with our Maieutix software in order to get, at the end of the day, some good amount of outputs. That is an unbiased binding site identification. So Maieutix can identify the binding site, can generate 3D ligand poses, so can give the different poses inside the binding site, can evaluate the non-covalent interactions inside this pocket, and can give the energy per residue values, and can compute the energy of all the complex target ligand complex. The simulation, the algorithm works as follows. So we will begin with an initial setup. So Maieutix defines the donors and acceptors in the hydrogen bond interaction, define the salinity, the temperature that will be kept constant all along the simulation. We define water in two ways, intrinsically and explicitly. So in intrinsic water is simulated with a modified Born approximation, and explicit water is defined by the water molecules by themselves. And Maieutix can define as well the centers of its peace-taking interactions because they consider the peace-taking interaction. And van der Waals and electrostatic interactions are simulated with an OPLS force field. Solvation and hydrophobicity instead are simulated with empirical and effective potentials. Protonation is changes all along the simulation, changes according to the pH we define, so the pH is constant. We change the protonation with another Monte Carlo loop that it is inside the main one, and well, and all this initial setup is included in this main Monte Carlo loop that does the following. So we move the protein, the drug, and the water randomly. So maybe if you in the audience there are people that works in the Monte Carlo or on dynamics, for sure they will know this process. So we change the conformation of the protein, drug, and water randomly. After that, we compute the energy of such configuration, of the new configuration, and we accept it or reject it according to the energy of the new configuration or according to a mathematical condition that depends on this energy of the new configuration we just computed. Well, what can we use Maieutix algorithm for? Well, first of all, we can use it to identify the binding site, as we said before. Well, actually, at the beginning, we place the ligand outside the protein. So Maieutix algorithm places it directly, so in a random place, and this ligand driven by the force field that we define finds the binding site properly. Well, here I present the case of a protein in which the binding pocket is not solvent exposed and is very narrow, and well, we think that it is a well test in order to see if Maieutix works or not. We normally use kinases or proteases in which the binding pocket is very solvent exposed, and for it is very easy to find the position, the binding pocket. In this case, it in theory should be more difficult for the ligand to find it. So here, for example, we see this is the output we would get from our simulation. In this case of the BRD4, well, we see that the ligand can find the binding pocket. This binding pocket is surrounded by these residues. So here, at the end of the day, you will have the different poses of the ligand inside the binding pocket. Here we see that in yellow the crystallographic structure and in blue the simulated ones. The results will be this position. I'm not sure if you can see the cursor, but however, this position, and then the Maieutix results here on the right, in which there are the non-covalent interactions between the ligand and the residues that surround it, and the different energies. And here on the bottom, we see that we can say the energy of the complex, as well as what's the interaction that stabilizes the most of the system. So another thing that, well, the goal of Maieutix, the first phase of our research, is to give a compounds ranking in order of affinity to our customers. So we ask our customer 1,000, 10,000 compounds. We run our algorithm, and at the end of the day, we have a list of compounds in order of affinity. This is our aim. And here I want to show you that, well, Maieutix can do a good job in these situations, and we think that it is very interesting in order to apply it in complex systems. The first example I want to show you is the first case study is the Pim-1 Kinase. Pim-1 Kinase belongs to the Pim kinase family that includes as well Pim-2 and Pim-3 with different level of tissue expression. And well, it is has a strong biometric interest in identifying the selective inhibitors. That's why we thought it would be very interesting to test Maieutix in that situation, because it should be very important, and it would see a good sign. Okay, here, for example, here we can see the results. Here on the right, we see a table with all the compounds and with their corresponding IC50 values ordered in order of affinity. And here we see the Maieutix score column. It is our score, and Vina score column. Why do we use Vina in this work? Well, because we think that Vina is one of the most used free softwares in this kind of field. So we wanted to compare ourselves with Vina to see if we can be better in these cases, in this type of researches. If you take a look, you see that, well, Maieutix score, ranks very well the order of affinity. This can be seen as well in the plot with the plot on the left, in which we see that the dot points in the IC50 versus Maieutix score, the dots are lying on this well-behaved line with a good correlation. Instead, Vina score, well, as you can see here, Vina doesn't reproduce very well this order. Actually, Vina is good at finding the good poses inside the binding site, but it is not good to order the different ligands in order of affinity. The second case study, the RET kinase. The RET kinase, the transmembrane receptor. Well, it's another kinase. It is interesting as well. There's a strong interest in these proteins, and we want to see if Maieutix can be useful in order to find good inhibitors, selective inhibitors in this kind of systems. Well, as we can see here, as before, well, here on the right, we have the compound number, its corresponding IC50, and the Maieutix score and the Vina score. Here as well as before, we see that the Maieutix score, the different dots in this plot, we see that, well, they are approximately on a line with a good correlation, 0.73. Meanwhile, Vina score is not so good at it, as for the reason we said before, actually. So here in this system of RET kinase, Maieutix gives good results. Well, the last case study, the Cathepsin G. Well, the Cathepsin G is not a kinase. Cathepsin G is a protease. We wanted to test Maieutix in another sort of proteins to see if it's good at finding good inhibitors. And well, let's see what happens. Here we see the table on the right with different names, AC50s, and the Maieutix score and the Vina score, and we see as before that Maieutix can reproduce very well that rank of affinity with a good correlation number, 0.80. So it is really good. Vina is not so good. Actually, it's not. They don't lie. The dots of Vina don't lie on an increasing line that it should be. They lie in a decreasing one. But well, the reason is what I said before. Okay, so as a conclusions, our Essential Dynamic Monte Carlo EDMC Simulation algorithm identifies quite well the binding pockets, identifies the target ligand non-covalent interactions and the different poses inside the binding site, compute the energetic contribution of the binding for each residue, as well as the energy of all the system, and it can rank the potential ligands. And well, one thing that I didn't say before is that, well, Maieutix can be run in a 4-processor PC, and well, it takes 3 hours to compute one compound. Our goal is to perform a simulation in 15 minutes. That is to simulate 100 molecules in 1 day in the near future. Well, as a sum up, the services of Maieutix, well, binding site identification. We see that Maieutix can rank very well the different affinities of the compounds. Admin PC profile can be studied as well. New target identification and lead optimization. And well, and thank you very much. If you have any, well, if you are interested in Maieutix, want to start a project with us, we are always open to projects with other companies or research groups. So please don't hesitate to contact us. This is our email address. And well, if you have any question, any comment, well, they are welcome. Thank you very much.